Welcome back to the Deep Dive. Today we are undertaking a fundamental and truly monumental task, charting the entire journey of life's information flow.
That's right. We're diving deep into Chapter 11 of Carp Cell and Molecular Biology, focusing on the central dogma, how that DNA blueprint gets translated into functional protein.
Our mission today is to guide you through every concept, every single mechanism, and all those pivotal experiments, and we're going to do it in the exact order the source material presents it.
Think of this as a complete molecular roadmap. We'll start with the very definition of a gene, move through transcription, all the extensive RNA processing, and go all the way to the precise mechanics of translation and even some cutting-edge applications.
But before we get into the nuts and bolts of the modern cell, we really have to acknowledge the great cosmic riddle of molecular biology.
The chicken and egg problem.
Exactly. DNA stores the code, but to do anything, replicate, transcribe, even repair itself, it needs complex protein enzymes.
But those enzymes can't exist without the precise information that's stored in the DNA, so where did it all start?
So how did the system get off the ground?
Well, the scientific consensus really leans toward the RNA world hypothesis. The idea is that early life relied on RNA for, well, everything.
Not just for information storage.
No, for the catalytic work, too, the stuff that proteins handle today. RNA was the original dual-function molecule, both the blueprint and the machine.
And what's so fascinating is that this isn't just a neat theory. We can actually see the fossil evidence of this RNA world.
Yes, in the most ancient and essential machinery of the cell.
The ribosome.
The ribosome, exactly. If you look at a high-resolution X-ray crystallography image of the large prokaryotic ribosomal subunit, you can see the catalytic center, the exact spot where the polypeptide chain is built.
And that spot, the active site, it's all RNA.
It's all RNA. The peptidyl transferase center. The proteins, which you'd expect to be doing the work, are sort of relegated to the outside, providing structural support.
So the RNA itself is the engine.
It is. It's probably the strongest piece of evidence we have for RNA's ancient catalytic role in the most basic function of life.
Okay, so let's start at the beginning. Phase 1, defining the gene.
We begin with the concept of the gene itself, which, has changed dramatically over the last century. It started with Mendel, who saw the gene as just a discrete element determining a trait.
Then Boveri and Sutton put those genes on chromosomes.
Yeah.
And later, Morgan showed they have specific addresses on those chromosomes.
Right. And then after that whole identity crisis, the work by Griffith, Avery, and the really famous Hershey and Chase experiment DNA was finally confirmed as the genetic material.
Which set the stage for Watson and Crick to define its structure. But all of that just defined what a gene is and where it lives.
It didn't explain what a gene actually does.
No.
And that revolutionary insight came much, much earlier from Archibald Garrod back in 1908. He was studying these hereditary diseases he called inborn errors of metabolism.
And he proposed this direct linear relationship between a gene and a metabolic product.
Exactly. The classic example from the source is alkaptonuria. Patients with this condition have urine that turns black when it's exposed to air.
And Garrod hypothesized that these individuals were just missing a specific enzyme.
Homogentic acid oxidase. It's what normally breaks down certain amino acids. So the logic was flawless. A defective gene led directly to a missing enzyme.
Which led directly to a specific metabolic problem. It established that genes encode function through enzymes, long before we knew anything about codons or polymerases.
Decades later, Garrod's ideas were resurrected by George Beadle and Edward Tatum in the 1940s.
Using the red bread mold. Neurospora.
Right. And their goal was to prove, experimentally, that one gene defect corresponded to one missing biochemical step.
So let's walk through their experiment, which is laid out so clearly in figure 11.1.
Okay, so first, step 1, they irradiated neurospore stores to induce mutations in the DNA.
Just randomly blasting the genome.
Exactly. Then, and this is the critical part, step 2, they grew these spores on a supplemented medium. This means the mold got all the nutrients it could possibly need.
So even if the damaged gene needed to make, say, a vitamin, the spore would survive because it was being fed that vitamin.
Precisely. Then came the screen. In step 3, they took these mutated spores and tried to grow them on minimal medium, a diet with no supplements.
And if it failed to grow on the minimal medium, but it grew just fine on the supplemented one.
They knew they had a nutritional mutant. They'd broken something essential.
And then came the really genius part, mapping the problem.
Steps 4 and 5. They would test that mutant on minimal media that was supplemented with just one single nutrient at a time. maybe just thiamine or just arginine.
So if the mold only grew when you gave it pantothenic acid,
You could deduce that the gene you broke was the one responsible for making the enzyme that synthesizes pantothenic acid,
it's so elegant. And that led to the one gene, one enzyme hypothesis.
which was later refined to one gene, one polypeptide, once we understood that many enzymes are made of several different polypeptide chains.
And today we'd even say one gene, one functional product, because so many genes just make functional RNAs. But the core idea holds.
The gene is the blueprint, and the process of using that blueprint is gene expression. The first step is transcription.
Making an RNA copy from the DNA template.
It's essentially rewriting the code from the DNA alphabet with T to the RNA alphabet with U, maintaining that complementarity. And the messenger molecule is, of course, messenger RNA or mRNA.
And the beauty of the eukaryotic system, which you can see in the overview in figure 11.2, is that separation. The master blueprint, the DNA, stays safe in the nucleus.
While these disposable mobile mRNA copies are sent out into the cytoplasm, it also allows for massive amplification. One gene can generate thousands of mRNAs.
And each of those can produce thousands of proteins.
And while mRNA gets all the glory, transcription also produces the other key players. Ribosomal RNAs, rRNAs, which form the ribosome, and transfer RNAs, tRNAs, the adapters.
Now, figure 11.3 reminds us that RNA structure is way more complex than the DNA double helix. Why is that folding so important?
That folding is what allows RNA to do complex, often catalytic jobs. It's a relic of the RNA world. It folds back on itself, forming these double helical stems and single-stranded loops.
And you even see non-standard Betty's pairs like GU.
Right, and modified bases, which are all vital for stabilizing these complex 3D shapes and for creating recognition sites for other molecules.
Okay, so let's get into the mechanics. The enzymes that do this are the DNA-dependent RNA polymerases.
First, the polymerase has to find the promoter site on the DNA. That's the start line. And crucially, these polymerases don't work alone. They need help from accessory proteins.
Transcription factors.
Transcription factors. Now, the mechanics of elongation, which you can see in figure 11.4, are very defined. The polymerase moves along the template strand from 3' to 5'.
And it builds the new RNA strand from 5' to 3'.
Right. And the chemistry here is engineered to be irreversible. The polymerase takes in a ribonucleoside triphosphate, an NTP, and cleaves off 2 phosphates.
We're releasing pyrophosphate or PPI.
But then a separate enzyme, pyrophosphatase, immediately hydrolyzes that PPI into 2 inorganic phosphates.
So the cell spends extra energy just to make sure the process can't go backward.
Exactly, it's a commitment device. That hydrolysis releases so much free energy that the whole reaction is driven forward.
And specificity is just standard Watson-Crick base pairing. The correct NTP fits and it gets added.
Right. And as it moves, the DNA unwinds in a bubble and then reforms behind the enzyme. These enzymes are also incredibly processive,
Meaning they stay attached for a very long time.
for long, long stretches of DNA, adding 20 to 50 nucleotides every second.
And this has been captured with these amazing single molecule techniques, which are summarized in figure 11.5.
They are incredible. In figure 11.5a, if you can picture it, researchers fix an RNA polymerase to a slide. Then they attach the DNA template to a fluorescent bead.
Okay, so as the polymerase pulls the DNA through to transcribe it,
The bead moves further away. You can literally watch it and measure the speed of transcription in real time.
and figure 11.5b takes it even further with optical traps.
Yes, these are focused laser beams that can hold that bead with a measurable force. By seeing how much force it took to stall the enzyme, they confirm that RNA pull is an extremely powerful molecular motor.
More powerful than even things like myosin.
Several times more powerful. But they also found that it's not a smooth ride. The enzyme pauses, it backtracks. If it makes a mistake, it has to go back, chew away the error, and then restart. It has a proofreading function.
So let's compare the machinery in bacteria versus eukaryotes.
The basics are conserved, but it's much simpler in bacteria. Prokaryotes have a single RNA polymerase called the core enzyme.
And by itself, the core enzyme is useless.
Completely, it just binds DNA randomly. The magic happens when an accessory protein called the sigma factor joins in, forming the holoenzyme.
The sigma factor is the guidance system.
It is. As you can see in figure 11.6, the holoenzyme now has a huge affinity for specific promoter sites and a much lower affinity for random DNA.
And those promoter sites have specific sequences, right? Figure 11.7 shows them.
2 consensus sequences, the nodic 35 element, which is about 35 base pairs upstream, and the Nigus 10 element, or the Pribnow box.
TTATAT.
Right, and it's the sigma factor that recognizes these, especially the Pribnow box, and that allows it to actually melt the DNA strands to form the open complex. It physically flips out nucleotides to start the process.
And bacteria use a whole suite of different sigma factors.
They do. Sigma 70 is the main housekeeping one for daily genes. But if the cell gets stressed, say by heat shock, it deploys a different sigma factor to turn on a whole coordinated set of defense genes.
And how do they stop?
2 ways. Either the Rho protein, which is this ring that zips along the new RNA and pries it off the DNA, or the RNA itself contains a terminator sequence that folds into a hairpin loop and causes the polymerase to just fall off.
In stark contrast, eukaryotic transcription is just layers of complexity.
It is. The first big difference is 3 different nuclear RNA polymerases, which are detailed in Table 11.1.
So we have Pol-1 for the big ribosomal RNAs.
Pol-1 is the star of the show, making the mRNAs and all the small regulatory RNAs.
And Pol-2 does the tRNAs and the little 5S RNA.
Exactly. And figure 11.8 shows this amazing structural comparison that reinforces evolutionary history. The archaeal and eukaryotic polymerases look much more similar to each other than either does to the bacterial one.
But the real complication in eukaryotes is the total reliance on general transcription factors, or GTFs.
Right. It's not just one sigma factor. It's a whole host of GTFs that are needed just to get Pol-2 to the promoter and get it started.
And the final key difference: nothing is ready to use off the production line.
No. The initial product, the primary transcript or pre-RNA, is much longer than the final molecule and needs extensive processing.
Okay, so let's get into that processing, starting with the stable RNAs.
We'll start with ribosomal RNA, which makes up over 80% of all the RNA in a cell. The demand is so high that the genes for it, the rDNA, are highly repetitive, hundreds of copies.
And they're all clustered in these specific nuclear structures, the nucleoli.
Right. Figure 11.10 shows them as these dense spheres in the nucleus. The nucleolus isn't an organelle with a membrane, it's just this dynamic, self-organized factory for making ribosomes.
And its internal structure, in 11.10ab, reflects that function.
It does. You have the fibrillar centers, the FCC, which contain the rDNA itself. Surrounding that is the dense fibrillar component, or DFC, which is where Pol-1 is actively transcribing.
And then the granular component is where the final assembly happens.
Exactly. So Pol-1 makes this huge 45S pre-RNA, which is the precursor for the 28S, 18S, and 5.8S rRNAs.
But wait, what about the fourth one, the 5S RNA?
That's made separately by Pol-3 outside the nucleolus and then imported for assembly.
So the 45S transcript gets processed.
Heavily processed. And the first thing that happens, even before it's cut, is 2 major modifications. Over 100 methyl groups are added to ribose sugars, and about 95 uridines are converted to pseudoridine.
And these modifications are targeted only to the parts that will become the final rRNAs.
Right, which implies they are critical for proper folding or function.
And this whole pathway was tracked using a pulse chase experiment, shown in figure 11.11.
Right. They use labeled methionine to track the methyl groups. In a short pulse, all the radioactivity shows up in the 45S peak. It's the first product.
Then during the chase, that 45S peak disappears.
And you see it move into intermediate products like the 41S and 32S precursors in the nucleolus.
What's really key for you to follow here is the timing. The small 18S RNA shows up really quickly in the cytoplasm. It's cleaved and exported fast.
While the other components take much longer to appear. And figure 11.12 shows that more than half of that original 45S transcript is just thrown away.
So how is all this trimming and modification guided?
Not by proteins alone, but by other RNAs. The small nucleolar RNAs are snorins.
Snornas. And they come in 2 flavors, right? As shown in figure 11.13.
2 main groups. The Box CD snorinase guide the methylation, and the Box HACA snorinase guide the pseudoridylation.
And their function is based entirely on RNA base pairing.
It's beautiful. The snoRNA has a sequence that's perfectly complementary to the spot on the pre-RNA that needs to be modified. It acts like a guide, bringing the modifying enzyme to the exact right nucleotide.
Incredible precision. Now, what about tRNAs?
They're also stable RNAs, made by RNA Pol-3. And interestingly, Pol-3 often binds to an internal promoter that's located right inside the gene it's transcribing.
Weird. And like RNA, the tRNA genes are also highly repetitive.
And their processing, shown in figure 11.14, involves trimming both ends, heavy modification of internal bases, and sometimes removing a small intron.
The book highlights one enzyme in particular, RNase P.
Yes, and this is historically significant. RNase P cleaves the 5' end of the pre-tRNA. And what Sidney Altman discovered is that while it has protein and RNA,
Catalytic activity, the actual cutting, is in the RNA subunit.
it's a true ribozyme, acting on a separate substrate. Another huge piece of evidence for the RNA world.
All right, that brings us to the main event, phase 3, building and splicing mRNA.
Yes, RNA Pol II and the synthesis of messenger RNA. The precursors were first identified as heterogeneous nuclear RNAs, or hnRNAs.
And the connection between these huge hnRNAs and the smaller final mRNAs was shown with another pulse chase experiment in figure 11.5 in it.
Right. Researchers gave cells a short pulse of radioactive uridine. The brand new RNA, the radioactive stuff, was all found in these enormous nuclear-only molecules, the hnRNAs.
But then during the chase,
The radioactivity in the hnRNAs vanished, and at the same time, it started showing up in the smaller, stable, cytoplasmic mRNAs.
a clear precursor product relationship.
Absolutely. So Pol II second synthesizes these pre-mRNAs with the help of the general transcription factors. The key recognition site for many genes is the TATA box.
AATA, about 25 bases upstream.
And the assembly of the pre-initiation complex, the PIC, is this really beautiful stepwise process. You can see it in figure 11.1 sting B.
It starts with TFIID, which contains the TATA binding protein, or TBP.
Right, and TBP binding actually bends the DNA, which helps recruit the other factors like TFIE, and then the polymerase itself comes in with TFI.
And it all culminates with TFIIH.
TFIIH is the key that turns the engine over. It has two jobs. It's a helicase, so it unwinds the DNA to make the transcription bubble.
And it's a kinase. It phosphorylates the polymerase.
Exactly. It phosphorylates the C-terminal domain, or CTD, of RNA Pol II, which is this long, repetitive tail.
And that phosphorylation is the trigger.
It is. As you see in figure 11.18, phosphorylation of one amino acid, serine 5, by TFIIH, is the go signal. It allows the polymerase to escape the promoter.
And then later, a different kinase phosphorylates another amino acid, serine 2.
Right. And this changing phosphorylation pattern is critical because the CTD acts as a dynamic scaffold. It's like a platform that recruits different processing enzymes at different stages of transcription.
It's an information board on the back of the polymerase.
A perfect way to put it.
So let's look at the structure of the final mature mRNA in figure 11.19. It's got the coding sequence, the UTRs, and two big modifications.
The 5' cap and the 3' polygal.
The 5' cap gets added almost immediately, right? Co-transcriptionally.
Yes. As shown in figure 11.26, enzymes add this inverted GMP residue with a unique 5' to 5' bridge. The cap is essential. It prevents degradation, helps with export, and crucially, it's the signal for translation to start.
And the 3' poly A tail is added at the end.
It's recognition-driven. The sequence AAUAA in the RNA signals a protein complex to bind, cleave the transcript, and then poly-A polymerase adds hundreds of adenosine residues without a template.
But the real bombshell discovery in 1977 was split genes, introns and exons.
A complete paradigm shift. Until then, everyone assumed genes were continuous stretches of code.
And the first clue came from studying adenovirus. Figure 11.21 shows how the 5' end of a viral mRNA was actually encoded by 3 totally separate segments of DNA.
Separated by these huge intervening sequences that were just gone from the final message.
And this was immediately confirmed in our own genes, like the globin gene mapping in figure 11.22.
Right. They compared the map of the genomic DNA to the map of cDNA, which is copied from the mature mRNA. And the genomic DNA had these massive regions that were completely absent from the final product.
The intervening sequences were named introns.
And the parts that are expressed that remain were named exons. And the staggering part is that introns can make up more than 95% of a human gene.
The physical proof came from R-loop visualization in figures 11.23 and 11.24.
This is a great visual. If you can imagine, you take the mature mRNA, which has no introns, and you try to hybridize it to the genomic DNA, which does have introns.
The exons will pair up perfectly.
But the part of the genomic DNA that corresponds to the intron has nothing to bind to, so it just bulges out as this big loop of double-stranded DNA, an R-loop.
And the image of the ovalbumin gene, figure 11.24, shows 7 distinct loops. Just stunning.
So this proved that introns are transcribed and then removed. a process we call RNA splicing. And it has to be absolutely precise.
Off by one nucleotide and you shift the entire reading frame.
Exactly. And that precision relies on consensus sequences at the splice sites, the 5' site, the 3' site, and an internal branch point adenosine.
And the mechanism here again points back to the RNA world.
It does. Figure 11.29 shows these self-splicing group 2 introns. They excise themselves using 2 chemical reactions, and they form this characteristic loop structure called a lariat intermediate.
And that exact same lariat mechanism is used for our pre-mRNAs.
But in our cells, it's not self-splicing. It's catalyzed by a massive machine called the spliceosome,
Which is made of proteins and 5 small nuclear ribonuclear proteins, the snRNPs.
SNRNPs, U1, U2, U4, U6, and U5.
Okay, let's walk through the assembly in figure 11.30 because it's a complex dance.
It is. It starts with the U1 snRNP binding to the 5' splice site, and U2 binds to that branch point adenosine. This basically defines the 2 ends of the intron.
Then the big U4, U6, U5 complex comes in.
Right, and this is where the RNA gymnastics happen. U4 is basically a chaperone for U6. U4 gets stripped away, which requires energy, and that allows U6 to base pair with U2.
And that U6-U2 pairing forms the catalytic core of the machine.
It does. U6 then displaces U1 at the 5' site, and the prevailing model is that the U6 snRNA itself acts as the ribozyme that performs the splicing chemistry. It's a dynamic RNA-based enzyme,
And again, the CTD of POL2 is the scaffold for this whole mRNA factory.
coordinating capping, splicing, and polyadenylation, all while the gene is still being transcribed.
So evolutionarily, what's the point of all this?
2 massive advantages. First, alternative splicing. A single gene can produce many different proteins just by splicing the exons together in different combinations.
And second, exon shuffling. Introns act like spacers that allow evolution to tinker, to move functional protein domains encoded by exons between different genes, creating new proteins much faster.
It may have allowed evolution to proceed by these quantum leaps.
Okay, so from splicing, we moved into the world of RNA regulation and genome defense.
Yes, the small regulatory RNAs. And the story starts, of all places, with petunias, figure 11.34a.
Right. Researchers tried to make petunias more purple by adding extra pigment genes.
And instead, the plants turned white. The extra genes somehow silenced both themselves and the original gene. They called it post-transcriptional gene silencing.
And the mechanism was figured out by Fire and Mello and C. elegans. The trigger was double-stranded RNA.
dsRNA. This was the birth of RNA interference, or RNAi. As you see in figure 11.34c, if you inject dsRNA that matches an mRNA, you destroy that message.
So the SIRNA pathway in figure 11.35a is our genomic immune system.
It is. It protects against viruses and transposons. The pathway starts when that foreign dsRNA is found and chopped up by an enzyme called dicer.
Into short interfering RNAs, or SIRNAs.
About 21 nucleotides long, these SIRNAs are then loaded into the RISC complex, which contains the key protein Argonaute or AGO2.
The passenger strand of the SIRNA gets discarded and the guide strand is left.
And that guide strand directs RISC to the complementary target mRNA. Once it binds, Argonaute acts like a pair of molecular scissors and cleaves the target. Destruction.
And the clinical potential here is just enormous.
It is. SIRNAs offer a way to specifically target and shut down any gene you want. The Human Perspective box talks about some early failures, mainly due to delivery problems.
But it also highlights the first FDA-approved SIRNA therapy, patisiran.
A huge success story. It treats a genetic disease, ATTR, where a misfolded protein builds up. The drug delivers SIRNA packaged in a lipid nanoparticle to the liver,
where it intercepts the faulty mRNA and stops the toxic protein from ever being made. It's revolutionary.
Now, we also have our own endogenous versions of these, microRNAs, or mRNAs.
Discovered in C. elegans, these are encoded by our own genome and regulate normal cellular processes.
Their synthesis pathway, in figure 11.35b, is a bit different. They're transcribed by POL2 as a hairpin structure.
And they get one cut in the nucleus by an enzyme called drosha.
Then they're exported, and then dicer makes the second cut.
But the key difference in how they work is the binding.
Right, exactly. When the mRNA in the RISC complex binds to its target mRNA, the pairing is usually imperfect. There's a bulge. This doesn't usually lead to cleavage, but rather to inhibition of translation or just general destabilization of the message.
So it's more about fine-tuning expression level.
Right. And one single mRNA can regulate hundreds of different mRNAs, allowing for these broad, coordinated shifts in gene expression, which you can see in the developing zebrafish in figure 11.36.
And we should briefly mention piRNAs.
Yes, piwi-interacting RNAs. They're longer, found in germ cells, and their main job is to suppress transposons to protect the genome of the next generation.
Plants take this a step further with long-range SERNI movement.
They do. They have an amplification loop. They use an enzyme called RNA-directed RNA polymerase to make more and more dsRNA from the initial trigger.
Which dicer then turns into more SERNAs.
And this massive wave of SERNAs can travel through the plant's vascular system, the phloem, creating systemic immunity to a virus. It's incredible.
And finally, we have CRISPR, the bacterial defense system that changed everything.
It's their adaptive immune system. The CRISPR locus in the bacterial genome contains little snippets of DNA from viruses that have attacked it before. It's a memory bank.
This memory is transcribed into a guide RNA,
which is then loaded onto the Cas9 protein. Cas9 uses that guide RNA to find and destroy matching viral DNA if it ever showed up again.
And of course, the revolutionary application is genome editing. We can now design a guide RNA for any gene we want and use Cas9 to make a precise cut, allowing us to edit the genome.
And the chapter ends this section by noting that a huge portion of our genome, maybe 2/3, is transcribed into long, non-coding RNAs and other things we used to call junk.
And the debate is whether this is just transcriptional noise.
Or a whole new, undiscovered layer of complex regulation. And given what we've learned about mRNAs and pRNAs, that second view is looking more and more likely.
Okay, phase 5. We have our mature mRNA. Now we translate it.
Right, decoding the information. Section 11.8 is about the genetic code itself. We know it had to be a triplet code, 3 bases per codon.
Because a 2-base code only gives you 16 combinations, and you need at least 20 for the amino acids.
And we know the code is non-overlapping. A single base change only affects one amino acid.
And it's degenerate. Most amino acids have more than one codon.
The code was cracked by Nirenberg and Mathai. Their big breakthrough was using synthetic poly-U RNA and showing it produced only polyphenyllanine. So UUU equals phenylalanine.
And that led to the whole decoder chart in figure 11.38.
And the code has these beautiful built-in protections. Degeneracy means a lot of single base changes are synonymous. The amino acid doesn't change at all.
And even when it is non-synonymous or missense, as you can see in the chart, codons for chemically similar amino acids are clustered together.
Right, so a mutation is more likely to swap in a similar amino acid, minimizing the damage.
Of course, the most destructive mutations are nonsense mutations, which create a premature stop codon.
And frameshift mutations and insertion or deletion, which scrambles the entire message downstream.
The key to decoding are the adapter molecules, the tRNAs.
These small RNAs, shown in figure 11.4, fold into this characteristic 2D cloverleaf, but their functional shape is a compact 3D L shape.
And the 2 business ends are the 3' end, where the amino acid attaches, and the anticodon loop on the other side.
And the reason we don't need 61 different tRNAs for the 61 sense codons is Crick's wobble hypothesis.
Base pairing is strict for the first 2 codon positions, but flexible at the third.
It's an efficiency hack. A single tRNA can recognize multiple synonymous codons, reducing the number of tRNAs the cell needs to make.
But before they can work, tRNAs have to be charged.
Yes, linked to the correct amino acid by enzymes called aminoacyl-tRNA synthetases. This is probably the most critical proofreading step in the whole process.
Figure 11.43 shows the synthetase recognizing its specific tRNA, and the charging is a 2-step, energy-requiring reaction.
Step 1 is activation. ATP and the amino acid form an aminoacyl AMP intermediate. That provides the energy. Step 2 is the transfer of that amino acid onto the tRNA.
And that high-energy bond between the amino acid and the tRNA is what will actually fuel peptide bond formation later on in the ribosome.
Exactly. And the synthetases also have an editing site to remove any incorrectly added amino acids, ensuring extreme accuracy.
So finally, we're at the ribosome. Let's translate.
First, the ribosome has to find the AUG start codon to set the reading frame. In bacteria, this is straightforward. The small subunit, as you can see in figure 11.44, recognizes the Shindall-Garneau sequence on the mRNA.
And that positions it perfectly at the AUG.
Right. But in eukaryotes, it's more complex. Figure 11.45 shows how the small subunit binds to the 5' cap of the mRNA first.
With the help of a ton of eukaryotic initiation factors, EIFs.
And then stands down the message until it finds the first good AUG. It's also fascinating that the mRNA is circularized during this process, with factors linking the 5' cap and the 3' poly A tail.
For efficiency, probably.
Most likely. Once the ribosome is assembled, it has 3 sites for tRNAs, as shown in figure 11.46, the A site for arrival, the P site for the peptide, and the E site for exit.
And the functional core of this machine is RNA.
It is. So the elongation cycle in figure 11.47 is a repetitive three-step process. Step 1 is selection. The correct charged tRNA, escorted by an elongation factor, enters the A site.
Step 2 is peptide bond formation. The growing polypeptide chain is transferred from the tRNA in the P site to the amino acid on the tRNA in the A site.
And this is the catalytic step performed by the ribosomal RNA, the ribozyme.
Step 3 is translocation. The ribosome moves exactly one codon down the mRNA, which requires another elongation factor in GTT.
This is a huge mechanical movement, or ratchet-like motion of the small subunit, as you see in figure 11.48. It shifts the tRNAs over, leaving the A site empty for the next cycle.
And the experimental pathways box really hammers home that RNA is the catalyst.
It does, citing Tom Seck's self-splicing RNA and Sid Altman's RNase P, but the key experiment was from Harry Noller.
He stripped almost all the protein off of ribosomes.
And the remaining RNA could still catalyze peptide bond formation. The final X-ray structures confirmed it. There are no proteins near the active site. It's an RNA machine.
The cycle ends with termination, figure 11.49, when a stop codon enters the A site.
No tRNA combined. Instead, a release factor binds, and it triggers the hydrolysis that cuts the finished polypeptide chain from the final tRNA.
Then we also have to mention the quality control system, nonsense-mediated decay, or NMD.
This is a surveillance system that finds and destroys mRNAs that have a premature stop codon.
How does it know it's premature?
By using landmarks left over from splicing, called exon junction complexes, or EJCs. Normally, the ribosome knocks these off as it translates.
But if it stops early, the downstream EJCs remain.
And those remaining EJCs are a signal to the cell. This message is faulty. Destroy it.
It's a way to prevent truncated, potentially toxic proteins from being made. And to maximize efficiency, you get polyribosomes, or polysomes.
Multiple ribosomes translating the same mRNA at once, as you see in figure 11.50.
Which brings us to figure 11.51, one of the most iconic images in molecular biology.
The coupling of transcription and translation in bacteria. Because there's no nucleus, ribosomes can jump onto the mRNA and start translating it even before the RNA polymerase has finished making it.
You're seeing the entire central dogma happening at once, in real time.
A beautiful, coordinated process.
So the chapter ends by pivoting to a cutting-edge application, DNA origami.
Right, using DNA not as information, but as a structural material.
The method, in figure 11.52, is brilliant. You start with a long, single-stranded scaffold, often from a virus.
And then you add hundreds of short staple strands. Each staple is designed to bind to two different parts of the scaffold, forcing that long strand to fold up into a precise, predefined shape.
And figure 11.53 shows the results, visualized with atomic force microscopy. You can make smiley faces, boxes, any 2D or 3D shape you can design.
And the applications are amazing, building tiny boxes for targeted drug delivery that only open at a disease site, or creating rigid scaffolds to arrange proteins to study how they work together.
We could even create DNA-templated electronic wires. It's incredible.
It's the future of nanotechnology.
So we have completed a massive deep dive into the central dogma, from the definition of the gene, the 5 classes of RNA, the spliceosome, RNAi, CRISPR, all the way to the ribosome.
And the most striking thing, really, is the dominance of RNA. It's the message, the adapter, the guide, and the catalyst. It's the legacy of the RNA world, front and center.
It really does feel like we've journeyed through the past, present, and future of molecular life in just this one chapter.
Indeed. And it makes you think. When you contrast the beautiful simplicity of that coupled transcription and translation in bacteria with the immense complexity of eukaryotic quality control, like NMD, it raises a question for you to mull over.
Okay.
Given how much energy the eukaryotic cell invests in surveillance, what other, maybe unlisted, super-fast mechanisms must exist right at the promoter to stop the production of faulty transcripts before they even get long? Especially if that crucial CTD phosphorylation platform somehow fails to recruit the right factors at the right time.
A great point on the need for redundancy in these high-stakes molecular factories. Thank you for joining us for this deep dive.
We'll see you next time.